Assalamu alaikum, guys, and good evening. Sorry for the delay. Uh, we are going to start from the point there we discussed last time. We were discussing about gene regulation, and we have completed the gene regulation in prokaryotes, and we studied about different operon models like the LEC operon and the TRP operon, and different types of the operon model with reference to the prokaryotes. So today we're going to start with the gene expression in the eukaryotes. We all know that the gene expression process in eukaryotes is much more complicated as compared to the prokaryotes, and definitely there will be certain stages where the gene regulation can be done in eukaryotes because the process itself is complicated and there are different levels at which the regulation can be done or different stages where the regulation can be done. So that's what I said, that the different organisms, they must regulate the gene expression at a particular time, and in multicellular organisms, this regulation of the gene expression is also essential for cell specialization. If you remember, last time I told you that for eukaryotic multicellular organisms, there is an extra step after the gene expression, which is involved in the cellular differentiation of cells based on their origin and based on their function. We'll not be discussing much about cellular differentiation at this stage, but just to let you know that it is also the part of the process of the gene expression to do the cellular differentiation. Now, almost all the cells of an organism are genetically identical, but they do have differences in their morphology. Like if you compare the morphology of a nephron with the neuron, and a neuron with the muscle cell, and the muscle cell with the cardiac cell, they would have differences in their morphology, and they do have differences in their functions as well. But when it comes to the genetic identity, almost all the cells of an organisms are pretty much genetically identical. Now, so if they are genetically identical, then what actually causes that difference between the cell type? So this is actually an important thing, that the differences between the cell types actually result from differential gene expression. That means the gene which are expressed in different kind of the cells are not same. Like if gene A is expressed in a particular cell line, then it might be possible that gene A will not be expressed in other cells, or it might be gene B or gene C. So this kind of differential gene expression actually in different cell types is the reason for the differences in terms of their functions, in terms of their morphology, etc. So the expression of the different genes by the cells with the same genome is actually the cause of this diversity. Now, abnormalities in different gene expression can also lead to certain disorders, certain diseases, which we call the genetic disorders, including cancer as one, kind of genetic disorder. So it shows that the gene expression is crucial and important with reference to the eukaryotic organisms as well, because as a result of the abnormalities of the gene expression, there is a chance of a genetic disorder, and in this case it could result in diseases like cancer and so on. Now, as I said, that in eukaryotes the gene expression is regulated at different stages because the process itself is complicated, and thereby there are stages or levels at which there would be expression of the gene regulation that takes place. If you look at this picture, it starts from this point where the first thing, that is the signal. Now, we all know that signal is a kind of stimulus. It could be internal stimulus or it could be external stimulus. For example, if this is the picture of a cell in front of you, which is in fact a picture of cell, the signal could be, let's say, carbohydrates, which are abundant in the blood. There's an increased level of the carbohydrate, that could be a signal, or there could be the decreased level of the carbohydrate, and that will also be considered as a signal. So any signal which is actually coming from outside the cell will have an impact on the nucleus. Now I'm just bypassing all the stages because you already have studied about how the signals are transmitted within the cells. Like, they have receptors on the cell surface, and then there are second messengers, and that second messengers will go further to deliver the message. So we're not talking about the details and how these signals are brought in, but always all kind of the gene regulations are being initiated by a signal, and that signal is actually for our genetic material, which is within the nucleus. So you can see the first step is the chromatin. As a result of that, the chromatin modification takes place, which may involve DNA unpacking, histone acetylation, and DNA demethylation. So there are three changes that could result as a signal approaches the cell or approaches the nucleus. So chromatin modification can take place, DNA unpacking will be done, or the histone acetylation is done, or the DNA demethylation is done. All these changes will have an impact on this whole process. Then the effect is on the DNA itself because the genes are coded on the DNA. And as a result of that stimuli, a particular gene is transcribed. That is transcription that you have studied. You got an RNA. The RNA that you got in the transcription, in case of the eukaryotes, if you remember, you call it a primary transcript because it will undergo some post-transcriptional modification. And we all have studied what these post-transcriptional modifications are. As a result of that RNA processing, you got the mature mRNA with a cap and a tail, and introns have been removed. And now this mature mRNA is going to transport out of the nucleus and will come into the cytoplasm, where it will be translated on the ribosomes. After the translation, you got a polypeptide, while the mRNA will be degraded. This polypeptide will then undergo different changes of processing, such as cleavage, chemical modification, etc., in order to give you the final active protein. Now, this active protein will then be transported to the site or the cell where it is needed. So let's say if it's an enzyme, this whole process starts with a signal, and that signal has been transformed by going through different stages to give you the final product, which is the protein, if it's an enzyme. And if this is no more needed, it will be degraded, but that's a different story. But if this protein is required in a particular cell, it will be transported to that particular destination where it is needed. So as I said earlier on, that if the signal is, let's say, increased level of sugar, increased level of sugar means the signal that we are talking about is insulin, and that insulin will have this impact on this whole process and ultimately would result in those enzymes which will make the glycogen from the glucose. That means the glucose that's coming into the cell will be stored as glycogen, and definitely you need enzymes for that. So that's how these are the stages, these are the levels in the picture showing you that at that levels or stages, the regulation of the gene expression can be done. Now, this is the same picture, a bit of a bigger portion to explain you what happened actually. As I said, signal impacting on the chromatin effect on the DNA, the DNA undergo transcription, and then RNA processing, and then uh, transport to the cytoplasm. And then from the lower on, the cytoplasm, their mRNA is translated to get the protein, and the protein will then be transformed into its active form. So these two pictures gives you an idea that how different stages will have an impact on the gene regulation in eukaryotes. We know that in eukaryotes, the DNA is not naked. Just like in case of the prokaryotes, it was a circular DNA, what we call the plasmid. But in case of the eukaryotes, the DNA are linear and much more complicated. And they are packed together with different proteins in the form of heterochromatin, which is usually not expressed. So actually genes which are packed within the heterochromatin are not expressed until unless you expose the genes, and then the process takes place. So how to unpack the heterochromatin in order to provide the genes the opportunity to express? There are certain stages like chemical modification to histone, because we know that in eukaryotes the DNA is wounded around the histone proteins for its support. So the chemical modification to histone and the DNA of the chromatin will influence both chromatin structure and gene expression. So we can say that the first stage of the regulation of the gene expression is at the level of chromatin structure, where the chemical modification to the histone, what we call the acetylation reaction, and the DNA, which we call the demethylation, will influence the gene expression. So this is the first stage or the first level of regulation in eukaryotes. So as I said, that histone play a very important role in providing the platform for the DNA to bond around, and that's how the supercoiling takes place. The modification that takes place in histone is called histone acetylation. Now, acetylation, is the addition of acetyl group. So in histone acetylation, an acetyl group is attached to the positively charged lysine amino acid of the histone tail. The result of this acetylation is that it loosens chromatin structure. Previously, the chromatin is tough and it has bound the genes within them. The histone acetylation results in losing of the chromatin structure, thereby permitting the initiation of transcription. So until unless this losing will not take place, there are no chances of transcription. So the first step is of the regulation is histone acetylation, which you have seen in the picture earlier on, which will come as a result of a stimuli, and then it exposes the DNA or the gene, and thereby promoting the initiation of transcription. Further, the addition of a methyl group called methylation will condense the chromatin. Like that's the reverse process. If a methyl group is added on the chromatin, instead of losing, it condenses. The addition of the phosphate group, that is phosphorylation, next to methylation amino acid can loosen chromatin. So these are the three things to understand, that the chemical modification of histone, either by acetylation or methylation and phosphorylation, can have the impact on the histones or the chromatin to make it loose or to make it more condensed. And that will definitely impact and regulate the process of gene regulation. This is a diagram showing you that these are the histone tails which comes out of the DNA molecule outward from the nucleosome, and that's the DNA double helix that is bonded around the histone. These are the amino acids available for chemical modification, and you can see that's how the modification takes place. This is actually unacetylated histone, and that is acetylated. You can see that this is more condensed, and as a result of the acetylation, it becomes loose and it's exposed. Once it is exposed, we say that it allows the process of transcription to start. So this is the first stage of regulation. The histone code hypothesis proposes that specific combination of modification as well as the order in which they occur will determine the chromatin configuration and influence transcription. So as you see that it's acetylation, methylation would have an inverse effect, and then phosphorylation. These all combinations of the modification and in the sequence in which they occur will affect the configuration of the chromatin and thereby affects the transcription process. The second modification is the DNA methylation. Now we have done the first stage. The first stage was the histone modification. The second stage is DNA methylation. The name itself indicates it's the addition of a methyl group to the DNA. Now the addition of a methyl group with a certain basis in the DNA is associated with reduced transcription in some species. So that means DNA methylation is a negative factor on the transcription in some species. Now, the DNA methylation can cause long-term inactivation of the genes in cellular differentiation process as well. In genomic imprinting, methylation regulates expression of either the maternal or paternal alleles of certain genes at the start of the development. So when we do the genomic imprinting of the newly born zygote baby, this methylation is being done as one of the techniques to regulate the expression of either maternal or paternal genes in order to diagnose and understand the genetic imprinting of the newly born zygote. There is a term called epigenetic inheritance. What is epigenetic? I'm sure you guys know what is genetic inheritance. Epi means above genetic. What actually epigenetic inheritance is? The chromatin modification we just discussed, like acetylation or methylation or phosphorylation, do not alter the DNA sequence. It just allows the chromatin to become more condensed or loose. But they may be passed to future generations of the cell. This is called epigenetic inheritance. Why we are calling it epigenetic? Because it's not altering the DNA. It's not genetic inheritance. Anything that will pass on to the DNA sequence will be kept in the genes. And if it's transmitted, we call it genetic inheritance. But this is not impacting the DNA sequence, it's impacting the chromatin modification, but it is transmissible from one generation to another generation, and that's why we call it epigenetic inheritance. The inheritance of the trait transmitted by mechanism not directly involving the nucleotide sequence is called as epigenetic inheritance. So I'm sure that you guys are clear on what is epigenetic inheritance. Now, the third stage of the regulation of the gene expression, and that the third stage is regulation of transcription initiation. Now, in this stage, the chromatin modifying enzyme, which provide initial control of the gene expression by making a region of DNA either more or less able to bind with the whole transcription machinery. We have already studied transcription. We know how the enzymes bind to the promoter region and then thereby causing a bubble to form the transcription fork, and then how it goes, we all have studied. So we know that chromatin modification is actually allowing that region to expose where the transcription machinery can bind and will start the transcription process. So the organization of a typical eukaryotic gene will play an important role in this. Now, associated with most of the eukaryotic genes, we have multiple control elements. That means, genes have got more than one control elements. And these elements are actually segments of non-coding DNA that serve as a binding site for transcription factors that help regulate transcription. Remember that in eukaryotic DNA, I told you that there are coding and non-coding sequences. So the non-coding sequences do not have the genes on them, but they do have the process, they do have the involvement in the process of the regulation of the transcription. So control elements and the transcription factors, they bind and they are critical to the precise regulation of the gene expression in different cell types. So there are certain control elements, certain molecules, certain proteins, I would say, they bind to the non-coding sequence of the DNA and thereby provide the basis of expression control in different cell types. Look at this diagram. You can see that this is the DNA molecule. We have got intron and exon, and we have proximal control elements here. We have promoter. This is the transcription start site. These are the enhancer. Now, these enhancers and proximal control unit, they are on the non-coding sequence, but they do have impact on this transcription start. And thereby, I'm saying that these, uh, these elements, control elements, can have the impact by binding to the non-coding region of the DNA and thereby impacting the transcription process. Now, here you can see as a result of this, we have the synthesis of primary RNA transcript, which we already have studied. And then the processing will take place and you got a mature mRNA molecule. Now, there are certain transcription factors, which are most of the time are proteins, and they do have their role in the transcription regulation as well. For example, to initiate transcription, the eukaryotic RNA polymerase need the assistance of certain proteins. We know that, the main enzyme doing the transcription in the eukaryotes is the RNA polymerase. And we know that RNA polymerase doesn't require a primer, if you remember last time we started. So this RNA polymerase will bind to the promoter region, or you can say the transcription start site, and then we all know the process. But the point is that the functioning of this eukaryotic RNA polymerase requires the assistance of some other proteins, and these proteins are being called as transcription factors. Now, these general transcription factors are essential for transcription of all genes, all protein coding genes. In eukaryotes, high levels of transcription of the particular genes depend on the control elements, which I mentioned earlier, interacting with specific transcription factors. So together with the control element and the transcription factors in eukaryotes, they are highly involved in the regulation of the transcription of particular genes. There are certain other proteins beside transcriptional factor. We call them enhancer and specific transcription factors, which I have already shown you in this picture. You can see the proximal control element and the enhancer, which are the distal control elements. So that's what I'm talking about here. The proximal control elements are located close to the promoter, while the distal control elements are the grouping of which are called enhancer, and they may be far away from the gene or even located in an intron region, as shown in this picture. There are activator, which are called activation protein or activator protein. They bind to the enhancer region, and as a result of the binding to the enhancer region, they stimulate the transcription of the gene. So we have certain proteins called activator. They bind on the enhancer region, which I just showed you, and that results in the stimulation of the transcription of the gene. The activators have two domains, one that binds with the DNA and the second that activates the transcription. Now, these bound activators, they facilitate a sequence of the protein-protein interaction that results in transcription of that particular given gene. So this, you can say that this is the DNA binding domain and this is the activation domain. So one side it binds to the DNA, the other side it binds to the enhancer region, and thereby it affects the process of gene expression or transcription. Some transcription factors function as repressors. In contrast to enhancers, we can have repressors, and the name itself indicates that they inhibit the expression of a particular gene, and they do have different methods to do that. Some activators and repressors, they act indirectly by not binding to the DNA, rather by influencing chromatin structure, earlier stage as I mentioned, to promote or silence transcription. So there are some proteins which, instead of impacting at the DNA level, can impact at the first stage where the chromatin was modified to either promote or to silence the transcription. So this is about the activator, this is about the enhancer. They bind with the enhancer, thereby triggers the promoter region, that is called the data box, and then the gene transcription would start. Here, the transcription factors bind. As a result of this, the process has been started. There are certain mediator protein that also play a role in this process, although we are not going in the detail for that. And then the RNA polymerase finally will have the chance to come and do the transcription. So before the RNA polymerase comes in, there are many specific proteins performing their role in initiation of the transcription process, which was not being seen in case of the prokaryotes, if you remember. Now, what is combinatorial control of gene activation? Combinatorial control of gene expression is a particular combination of different control elements that can activate transcription only when the appropriate activator proteins are present. That's why we call it combinatorial. Like the presence of certain control element will impact the activator proteins, which in turn enhance the process of gene transcription. That's why we call it combinatorial control of gene expression or gene activation, where two different molecules will have a combined effect. Now here comes the picture to explain. This is a liver cell and this is an eye cell. Now here we have the enhancer, we have the promoter, and then we have the albumin gene. And again, these are the control elements acting on this and this as well. The gene is the same, and you can see that the gene is the albumin gene, and here we have the gene what is called the crystalline gene. Now, in case of the liver cells, in the liver cell nucleus, there are available activators which will bring about the binding of the co-activators and then thereby facilitates the RNA polymerase and thereby facilitates the transcription of the albumin gene, and then it inhibits the crystalline gene as well. While in this case, the available activators will inhibit the expression of the albumin gene but enhance the activation of the crystalline gene. So in that case, one can say that this is what we call the combinatorial effect, where the available activators are working as a repressor in one cell and as an activator in the other cell, depending upon the need of the cell, which protein they actually need. This is the same picture, it's just being shown with a bigger picture of the liver cell, and that is the lens cell. Okay, let's go further. There is another stage of the regulation. And now this stage of regulation is at the post-transcriptional stage. We have primary RNA transcript produced. And now this primary RNA transcript will undergo post-transcriptional modification, which include 5' capping, 3' poly A tail, and removal of the intron. So the next stage of the regulation of the gene expression is at the post-transcriptional stage. So the transcription alone does not account for gene expression. The regulatory mechanisms can operate at various other stages after transcription. And such mechanisms allow a cell to fine-tune a gene, because the gene has been transcribed, but we need to fine-tune the gene expression, and that can be done after the transcription process, and that is why we call it the gene regulation at post-transcriptional stage. Now, in RNA processing, in alternative RNA splicing, different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exon and which are treated as intron. Oh, that's a very, very important point. What you have been told earlier on, that when the transcription takes place, we have both intron and exons, and we call the introns as the non-coding region and exons as the coding region, if you remember. But according to this RNA processing, it says that an intron can be an intron for particular protein, but it could be an exon for another protein. That means the code that has been transcribed on the primary mRNA may be treated as exon or intron depending upon which protein we are looking for. That's what we call the fine tuning. So it's not always like, the primary transcript RNA forms and always the intron and exons are same. No, it depends which protein we are interested in and which gene we have actually transcribed. Depending upon that, there is splicing, what we call alternative RNA splicing, where specific intron and exons are being treated as intron and exon to get the final product out of it. This is what we call RNA processing. So here you can see, let's take this example. This is a DNA molecule, and this is for the troponin T gene. So you have one, two, three, four, five. These are the exons. That means they must be present in the mature RNA molecule. Now you got the primary transcript. Now the primary transcript has got the one, two, three, four, five genes. Now the RNA splicing takes place, and now there are two mode of splicing. Now you can see there are two mRNA produced here. One mRNA has got gene 1, 2, 3, 5, and the other have 1, 2, 4, 5. Now both of these mRNA are different, and both of them are post-transcriptional modified mRNA. That means this will code for one protein and this will code for another protein. So RNA splicing is not only removal of the intron, but it is also involving which part of the RNA will be considered as intron and which part of the RNA would be considered as exon. So this is another level of gene regulation. Like for example, if let's say gene 4 is not required, so the RNA splicing takes place in a way that we don't have gene 4 here. Or if gene 3 is not required, or if we don't want to express, then the splicing takes place in a way that it has been removed. So this is called the fine tuning of the protein, or in other words, the gene regulation at the level of post transcriptional modification. Now, after all that, when the RNA is being read, we all know that this mRNA will be degraded because the half-life of the mRNA is short. Once you have the translation done, you got the protein, this mRNA is going to be degraded. So the lifespan of the mRNA molecule in the cytoplasm is a key to determine the protein synthesis. The eukaryotic mRNA is more long-lived than the prokaryotic mRNA. The nucleotide sequences that influence the lifespan of the mRNA in case of the eukaryotes resides in the untranslated region, which is called, I told you earlier on, the untranslated region is called the UTR region, which is at the 3' end of the molecule. So that is actually causing the stability, as the poly A tail we all know is mainly responsible for the stability of the mRNA molecule. Now the next step is the translation, and this is the next stage of the gene regulation. Let me summarize actually. We said that there are many stages of gene regulation. So we started with the first stage, which was the chromatin modification. Then we started about second stage, which is the DNA methylation. Then we studied about the third stage, which is the initiation of transcription. Then we have the fourth stage, which was initiation. Then we have the fifth stage, which is called, transcription. The fifth stage, we have the post-transcription. And now the sixth stage of the regulation is at the level of translation. And what we're talking about is here is initiation of translation. Now, the initiation of translation of a selected mRNA can be blocked by regulatory proteins. Like at this stage, for example, we have got mature mRNA now. At this stage, if we don't want that protein, that means we have to stop the translation process. And that translation process can be stopped by certain regulatory proteins. These regulatory proteins will bind to the sequences of the mRNA and thereby prevents the binding of the mRNA with the ribosome. And there how the protein synthesis is stopped. Alternatively, translation of all mRNA in a cell may be regulated simultaneously as well. For example, translation initiation factors, which are certain proteins which are required for the translation process, can activate or inactivate a particular gene. Like, as I said, the translation initiation factors are simultaneously activated in an egg following fertilization, because that's how we need the activation of all the genes which are being transcribed. Similarly, it can be reversed for that. The translation initiation factors can inactivate as well the process where the whole process is being switched off. So that means the gene regulation stage can also be set at the level of initiation of translation. The next stage, if the translation takes place, what you will get? You will get the primary protein structure, which needs processing, which needs modification in order to transform that into an active protein. We called it post translational modification. Now, this stage is also being part of the gene regulation, and the regulation can be done at this stage. After translation, you got different protein chains. They undergo breakdown, cleavage, addition of chemical groups, conformational change, and so on and so forth. The proteosomes are the giant protein complex that bind protein molecule and degrade them. That means you got the protein, it has been processed, you know, modified, but this final protein may attach with a giant protein called proteosomes, which results in the degradation of that protein. So you can see that how we started from the chromatin level and till the last stage where we got the final protein, that protein can be degraded if it is not needed. So these are the different levels where the regulation of the gene expression can be done. This is the example. You can see that this is a protein, which is the final product of the translation. The molecule of ubiquitin comes in, they bind with this, ubiquitinated protein. Then the proteasome comes in, and proteasomes bind with this and results in the degradation of the protein, and the protein is no more functional. So, with this, we have completed the entire regulation of the gene expression in eukaryotes. And I'm sure it's a little bit complicated, but go through it slowly and you will understand the different levels. So, if I summarize, I can say that the regulation is done at the level of the chromatin, the DNA methylation, then transcription, then post transcription, then translation, then post translation. These are the stages where the gene regulation takes place. Finally, we are going to discuss something about the disease conditions which actually result from the disturbances in the process of the gene regulation. If somehow the gene regulation is disturbed, if somehow the gene regulation is imbalanced, it would result in some sort of genetic disorders, and the most common of them, is the cancer, where we have the uncontrolled growth of the cells. So we are going to discuss a little bit about cancer results from the genetic changes. It actually changes the cell cycle control. If you remember the cell cycle, I told you in the very first lecture of Introduction to Molecular Genetics. So the gene regulation systems that go wrong during cancer are the very same systems involved in the embryonic development. So in this case, there are types of genes which are associated with specifically cancer. The cancer can be caused by mutation. Any mutation to the gene which is going to impact the control of the cell growth and division. And as a result of this, when the control of the cell division and growth is affected, that results in uncontrolled cell multiplication, uncontrolled cell division, and that is what actually results in cancer. There are certain viruses we call the tumor viruses, which are a reason for this mutation, and they cause cancer in animals, in humans, and in other species as well. These genes which are associated with cancer are being called as oncogenes. Oncogenes are the genes which are called cancer causing genes. There are genes called proto-oncogenes. These are the corresponding normal cellular genes which are responsible for the normal cell growth and division. So proto-oncogenes are the normal genes, oncogenes are the disturbed genes as a result of the mutation. The conversion of proto-oncogene to an oncogene results in abnormal stimulus of the cell cycle, in turn abnormal cell division and overgrowth of the cells. This is a proto-oncogene on a DNA molecule. As you can see that as a result of this proto-oncogene, the gene amplification takes place and you got multiple copies, and then the normal growth stimulus takes place. While in case of the point mutation of this proto-oncogene, the point mutation results that there is a change of this proto-oncogene to oncogenes. And now these oncogenes will be giving hyperactive or degradation resident protein, which would cause the cancer. While in case of the translocation sort of mutation, it results in the normal growth stimulating protein in excess. So this is how we see that we studied that not all mutations are dangerous, if you remember. So some mutations can result in the conversion of proto-oncogenes to oncogenes, but in some cases it will remain normal. The proto-oncogenes can be converted to oncogenes by following possibilities. Number one, movement of DNA within the genome. If it ends up near an active promoter, transcription may increase, and as a result of this, enhanced protein is produced, enhanced growth factors are produced, and that will result in cancer. Secondly, amplification of a proto-oncogene. Proto-oncogene is a normal gene. If it's amplified by some reason, it will increase the number of copies of the gene, and that is causing the cancer. A point mutation of a proto-oncogene can also result in the disturbance, and that causes an increase in the gene expression. So there could be different possibilities by which the proto-oncogenes can be converted to oncogenes and thereby causing cancer. Beside the oncogenes, we have certain genes, we call them tumor suppressor genes, and their function is opposite to the oncogenes. These genes are actually there to suppress the tumor genes, and they help prevent the uncontrolled cell growth. The mutations that decrease protein product of the tumor suppressor genes may contribute to cancer onset. Like if in our body, these tumor suppressor genes should be normally produced. If for some reason our body is producing less amount of tumor suppressor proteins or less expression of tumor suppressor genes, it ultimately contributes towards the cancer onset. So tumor repressor proteins are involved in repair damaging DNA, control cell adhesion, and inhibit the cell cycle in a cell signaling pathway. So any decrease in the tumor suppressor protein will contribute towards the cancer. This is another aspect of cancer production. Now, how it interferes with the normal cell signaling pathway? One example I'm giving you here. Mutations in the RAS proto-oncogene, this is one of the proto-oncogene, and P53, which is the most common tumor suppressor gene, are the most common in human cancers. So we have RAS proto-oncogene and P53. RAS proto-oncogene is an oncogene. The mutation of that results in cancer. And P53 is a tumor suppressor gene, and the inactivation of that gene also causes the human cancer. So these are the two most common reasons of the human cancer. The mutation in the RAS gene can lead to production of a hyperactive cell and increased cell division, while suppression of the P53 tumor repressor gene will not be able to control this RAS gene, and that's how the cancer progresses. So this is the pictorial representation. We can say this: the growth factors are there, and the receptors and the protein kinases. This is the mutation where the hyperactive RAS protein products of oncogenes issue signals, and as a result of this, it stimulates the process. So this is one impact. And you can go and discuss the cell cycle inhibiting pathway as well, like how they inhibit the cell cycle pathway. This is the P53 gene, which is actually being there to suppress that. But the defective or the missing P53 will be inactive, and it's not called doing its function, and that's how the uncontrolled growth goes on and on. And finally, you can see that cell cycles are not inhibited, and that's how the cancer progresses. This is the same picture again being shown in different parts. So finally, we end up today's lecture here that the suppression of the cell cycle is important in case of the damage to the DNA. The P53 prevents a cell from passing on mutation due to DNA damage, because P53 is involved in the DNA repair. So the mutations in the P53 gene prevent suppression of the cell cycle. That's why the most common gene of interest nowadays we study for the cancer treatment worldwide is that P53 gene, which is the most common gene of interest for cancer treatment. So we are going to stop here, and we have finished the lecture here. And if you have any question, let me know. Just raise your hand. Okay, so if there are no questions, I'm going to stop the lecture here, and I'll share the recording once it is available. And meanwhile, you can go through the presentation and come back to me for any question if you have. Thank you very much, and Khuda Hafiz.